Okay, we may as well get started. This is going to be a very brief session, so it's really just meant to get us started on the workshop for tomorrow. Just an overview of what I'm going to talk about and an introduction both to myself and to MPMAP a little bit in terms of making sure it's all working properly, talking about how we get data into the package, and then just a few simple exercises at the end. So I'm happy to be interrupted as I'm going through it. I think we've got plenty of time today. Tomorrow, this will at least have us getting off to a running start, basically, so we can get right into some of the more interesting bits of linkage map construction and QTL mapping in multi-parental populations. So the plan is basically today to go through an overview. So as I said, introduction, background, talking about installation. It should already all be installed on your computers, so hopefully that's not a problem. Talking a little bit about the documentation and importing data. So that's kind of the main thing. If any of you have multi-parental data that you're thinking of looking at, then feel free to ask me questions at the end, because if we can get the data into the right format today, then tomorrow it'll make it much easier to play with it as we're going through the package. Tomorrow then, the first hour we'll be talking about linkage map construction, and so I've set aside about 45 minutes, going through various steps. First, estimating recombination fractions. Second, grouping the markers together into linkage groups. Third, ordering markers within linkage groups, and then doing some refinement to try and improve your map, doing some diagnostics to identify where there might be problems with it. After that, we'll talk about QTL mapping, what we might want to do with the map, and luckily Carl has gone through quite a bit of it today, so you can build on that. Basically, I'm not going to go through what QTL mapping is or anything. I'm going to talk about how it might differ for a multi-parental population and why MPMAP might be useful in looking at different models that I talked about yesterday during my seminar. So the full model, modeling different effects for each of the parents, association mapping, just looking at the effects of the observed alleles, grouping different founders together to try and reduce the dimension of the problem, and why we might want to fit mixed models if we have more complicated phenotypes. And then the last hour is what I'm calling advanced topics, which are some things which have really, when I first developed MPMAP, it was really aimed at those two topics, linkage map construction and QTL mapping. And since then, various different modules have kind of been built onto MPMAP in each of these different areas, or they've been built into different R packages. And so I thought, either now, if people have an idea of which of the topics they'd like me to talk about, then I can already select them now, or at the end of the first couple sessions tomorrow, people can say, look, I would really like to hear more about visualization. How do I plot if I find interactions on my different chromosomes? How do I make things which are publication quality? Or if you're more interested in recombination, how can we use the haplotype mosaics in our populations to detect where recombination events are occurring? How do we map QTL for recombination events? And so on. So each of those 5 topics I could probably talk about for half an hour to an hour, but we'll pick a few of them, and I'll talk about each of them a little bit, show you how to do each of them in MPMAP, and then the rest of the information will be there in the slides anyway, so if you want to go through it separately on the topics we don't cover, then you can do that later on. So, as I said, this is an overview. When I first started building MPMAP, that was back in 2009, and the picture on the right is what I felt like it looked like after a while, where we had just sort of ended up tacking on extra functions. We'd started off really focused on linkage map construction and QTL mapping. We started tacking on extra functions. Multiple people were working on it. Some things were written in one person's style, some things were written in another person's style, and I've tried to clean it up lately to make it look more like the thing on the left. And what I'll do as I go through this workshop is to try and focus on the cleaned up parts. There are certainly a lot of functions in there which can be used to do lots of extra stuff, but as I said, these are things which have been put in at various times because somebody wanted to do something, and so while it might be useful, it's not something that you want to use on an everyday basis, I would say. Seth gave an introduction to my background yesterday. My background is definitely on the mathematical side. I'm not a computer programmer. I'm I have used R now for about 8 years, I guess, since I finished my PhD. And when I first started off, I wrote an R package during my postdoc. I went back to that R package about 2 or 3 years after I finished it, and I looked at my code and I thought, you know, I know many better ways to do this now. So as you go on, you certainly learn better ways of doing things, faster ways of doing things, and we try and update NPMap to accommodate that as we go through it, but there are parts of it which are slower, and I'll try and identify that for you as I go through it. So as I said, at CSIRO, 2009 is when I started working with the MAGIC populations. Then the focus was really highly on linkage map construction. We didn't have a linkage map for the population. We didn't have any sort of reference genome for wheat. And so when I came on, that was my mandate, basically come up with some way to construct linkage maps and ideally make it into a software package so that other people can use it. And so in 2011, we first published NPMap, but it looks very different from that form today. So there's an old version of it which is actually on CRAN, and there's a newer version which is updated regularly and is on GitHub. And I'll show you something about the installation later, actually. The one which is installed on the computer should be the one on GitHub, so that should not be a problem. In 2013, we held a workshop at the National Institute of Agricultural Botany in England on MAGIC-type populations, and I've put the link there to the slides from the slides from the workshop, if anyone's interested. That was mainly focused on bringing together people working in different crops and different model organisms with MAGIC populations and trying to share what their experiences have been. So there's a number of talks from people describing their populations, describing what their goals are and what kind of methods they've used. And then there are a number of people who are talking more about the types of methods you can use in those populations. Many of those were then published in a special collection of articles in Genetics and G3 in 2014. And it looks like the link got cut off there, but basically it says populations at the end. And then in 2015, there was another workshop at Plant and Animal Genome Asia on multi-parental populations. I don't think the talks are available for that online, but that was kind of in the same vein as these other workshops. So through meeting people at these workshops and through NPMap, I've actually collaborated with a number of people in different crops, in wheat, at CSIRO, in durum wheat, in barley, tomato, Arabidopsis, chickpea, and rice. And so there are a number of people working on MAGIC populations. If you're interested in one of these crops and you're interested in multi-parental populations, I encourage you to get in touch with them. Most of these have been published at this point. But in the future, going forward, I'm going to have much less involvement with MAGIC. So I did mention I'm moving on to a position in human medicine. And so I will be doing less in terms of maintenance and updating NPMap, but there are people at CSIRO who will be taking this on instead. I am continuing some involvement, though, so I'm actually presenting next week in the Netherlands on things related to MAGIC populations, and the same in China in November. So there's still going to be certainly some involvement as far as MAGIC and NPMap goes, so feel free to contact me by email if you have questions after this. Okay, so getting to the background of NPMap then, well, the question that might come up is why would you want to use it? There are some alternatives. So when I first started looking into this problem, part of what I did was to say, okay, well, what are other people doing? So at that time, the Arabidopsis MAGIC population had been published and the collaborative cross in mice had been published. And Carl was involved actually in the collaborative cross. So there's some functionality in RQTL, but he did mention that it's not really intended for multi-parental populations. Happy was also another software that was used in the collaborative cross. And the advantage of Happy is that it's applicable to more general designs. So you don't have to have a very well-structured 4-way or 8-way population to use Happy. It doesn't require a pedigree. It can be used on just about any design, but it's not intended for linkage map construction. It was intended for Arabidopsis and for mice, and in neither of those cases are they worried about constructing a linkage map at this point. So the same went for RQTL. There was some functionality for MAGIC populations, but not a huge amount when we were looking into it, and so that was why I decided to actually write our own R package in order to analyze our magic populations. And so MPMAP, because these already existed, it actually interfaces with both of them. If you're interested in comparing them, it's possible to do that within MPMAP. But it was really aimed to be more specific, focusing just on magic populations. So it was intended originally as an internal type of software package, and then it gradually expanded. But it's also more general in that it has a broad range of tools for linkage map construction and QTL mapping, and as it turns out, imputation and various other things as well. So in terms of installation, I mentioned that the most recent version is on GitHub. The easiest way to make sure you have the most up-to-date version is to actually use a package called DevTools and to install directly from GitHub. And this works quite well. I'm assuming that's how it got on all these computers. I checked to make sure that it was the most recent version earlier today, and that's the type of output that you'll get from loading it in. So you shouldn't actually need to run this. It will work. It will take a bit of time because there's C code in the package, and installing it will recompile all of that C code. So just be warned that that's what will happen. As far as documentation goes, I highly recommend looking at it. This is how I learned R, basically. So I had programmed in C and C++ before, and I started using R when I started my postdoc, and basically for the first year, I just looked up functions all the time to figure out what was going on. There's so many functions in R, there's so many packages. Reading the documentation is the easiest way to learn and to just try things out. So this should be the first stop for looking what kind of inputs you need for functions, what kind of outputs it'll give you, and related pages. So if you do one type of analysis, what might be the next step in your analysis? Where can you find some examples and so on? And so I've put examples there. In the case of MPMAP, it's set up so if you just do $MPMAP, it'll pull up sort of an overview page about the package. It'll tell you about some of the functions which are there for some of the basic analyses. And then two of the other functions there, read.mpcross is for actually reading data into it, and mpcross itself is a constructor function. So that gives you an idea of the sort of basic object that you're going to be operating on with MPMAP. I should say that we basically modeled a lot of it. Yes? Sorry? Oh, make it bigger? So I'm not sure. No, that's not going to work. I've opened it from R itself. So it's not going wider. I guess I could zoom in. I don't think that's going to help, actually, that one. Let me just minimize this. So let's go back to here. If I can't get it to go full screen today, I'll make sure I can do it tomorrow. So yeah, I apologize for that. I would have thought I'd be able to zoom in on this, but no. See, it's just affecting the RStudio page there. Yeah, so these slides were written in RStudio using R Markdown so that the code would show up well, but it looks like a side effect of that is that they are not displaying well. So I'll convert them to PDF for tomorrow. So yeah, so those are the functions basically for getting data into it, into MPMAP, and the basic object we use is called an mpcross object, so just like the cross objects in RQTL. And basically you can import data using files that you create or using objects which are existing in R already. The necessary components are having the genotypes of your progeny, so your inbred lines, the genotypes of your parents, which will be in a separate matrix, the pedigree, which is recording all the crosses used in the design, and then 2 vectors of indices. One which is just an index of the parental rows in the pedigree, which is typically from 1 to the number of parents, and another one which is the indices of the rows in the pedigree of the genotyped finals. You can also add on components, and this is what we do as we do more analyses. We add on components which are representing the genetic map, which are representing the phenotypes, which are representing genotype probabilities, and the actual results from QTL mapping. Now, importing data can be one of the most annoying parts of doing the analysis, actually, and this is where I usually get contacted. So there are a number of common issues I've run into in terms of helping other people importing data and in terms of even just trying to get the functions to work myself. One, the simplest one is that we require numeric genotypes. So I know in RQTL you could put in different characters for your genotypes and then just make sure you identify them. We actually require numbers, and that will cause problems. It will read in the data, but you won't be able to do any sort of analysis on it. It can be any number as long as it's consistent. So it doesn't have to be 0, 1, 2 or anything like that. Any sorts of numbers just identifying different alleles, as long as they're consistent between the parents and the progeny, are fine. The second area people tend to run into problems is the pedigree structure. So down at the bottom there are some examples of different ways I've seen that people have stored their pedigree information. And typically this is because, so these are all for 8-parent crosses. And essentially what you're storing is a record of which parents you were crossing together at which generation. You're probably recording, you may be recording how many generations it's been selfed and so on. So in that first one, in the first generation you cross together A, B, C, D, E, F, G, H. Then you cross together A, B, and C, D, and E, F, G, H. And then you cross all of them together, and then you self it for 5 generations. The same sort of thing is true in the second one. That's how it's set up. But the slashes now are differentiating different generations. And then the third one is just saying, okay, this is the order in which they were crossed. Assume that the generations make sense, basically, in terms of describing it. But none of these is how we set up the pedigree file. So these all have to be converted into the kind of structure that we use for our pedigree. And then the third problem is the indices, and I'll show you an example of that in a second. So in terms of trying to figure out how to get your data into MPMAP, the easiest thing is actually to look at some simulated data and make sure that what you're putting in matches the structure in that simulated data. So there's an example here which is in the documentation for MP-Cross and for sim.mpcross, which is basically generating a map where you have one chromosome of length 100 centimorgans. You have 51 markers equally spaced, so every 2 centimorgans. You're generating a pedigree, and in this case I'm just generating a 4-way pedigree with 2 individuals, so something very simple so I can display it on the screen. And then you're using this sim.mpcross function to generate an MP-Cross object. And if you can see it, then these are the different components of an MP-Cross object. You have your founder genotypes, you have your final genotypes. In this case, because it's simulated, you actually have information about which alleles are the same, identical by descent. You can have phenotype information, you can have map information, you have the pedigree, and then you have those 2 vectors of indices I mentioned. And in this case, if you're simulating it, you can have QTL genotypes as well, maintained separately. So your founder data is basically just a matrix where every row is one of the founders. So you would have 4 or 8 rows, and you'd have all of the marker information going across in the columns. It's very simple. And the same is true of the file that you'd input. It's basically, if you were to generate that matrix in R and output it to a file, that's what you'd expect the file to look like. The same is true of the final data. So you have all of your lines going down the rows, you have all of your markers going across. But the pedigree is set up so that basically you have one row for each of the individuals that were ever created throughout the generation of your population. So these first 4 rows indicate the founders in this population. You have 4 individuals. The parents, the male and female parents, are both set to 0 because we don't know where they came from. We then cross them together. So in the first generation, we cross parents 1 and 2 together and parents 3 and 4. In the second generation, we cross 5 and 6 together, and we do it twice. So we generate 2 individuals, both with parents 5 and 6. And then for each of those 2 individuals, we self them for 3 generations. So here you can see individual 9 is created by selfing individual 7. Individual 10 is created by selfing individual 9. And we go down. And then individual 11 is actually genotyped. So that's your recombinant inbred line. You've only selfed it for 3 generations, but this is just a simulated example. And that's what the pedigree should look like, essentially. So it ends up being quite a large file if you have a large number of selfings, but that it gets compressed later on, essentially. So, yep.
So if you did random mating,
yes.
With parents you didn't know male or female,
Mm-hmm.
that came fairly later in the process. Is that where you should start since you have no information before that point?
So if you did random mating, but it's recorded what happened, or do you not know?
Yeah, you wouldn't know which ones are male or female.
So the male or female information we don't use to a huge extent. So that's not a big problem. It would only be a problem if you wanted to keep track of that for some reason. But in terms of using the pedigree, which one is male and which one is female doesn't matter too much.
No, so if you have, like, if you think about an open pollinator population, you don't keep track of any of them.
Of any of them, yeah.
So is that your first generation you would put in, or would you put the founding parents in there before that?
So that would be a slightly different structure, probably. So you're starting from a limited set of parents and then you're just doing random mating for a while?
Yeah.
Yeah, and not retaining any of the information about the matings. And then you're selfing the individuals. Is that right?
Yeah, I'm just trying to envision, like, you have zeros there because that was the very first set of material you
Right.
used. If you have no information after that to some point,
Yeah.
then you just want to start with the first place you have information from?
So in this case, I would say you'd put the parents in. In this case, I wouldn't use most of the functions for that. So the reason we keep this information is because a lot of the information, a lot of the probabilities we calculate are based on how things are crossed together. And if you don't know that, those probabilities are not going to be quite correct. So I would actually recommend using something like HAPPY in that case, where they don't make an assumption about the structure of the pedigree. And so in that case, their probabilities tend to be a bit more variable than if you know the structure, but if you don't know it, then it's more robust.